Okay, so now another very common or X-linked pattern is X recessive inheritance. And here, this individual is affected and is carrying the mutation. But if you look at this person's offspring, no one among the children here are expressing the disease. And the reason for this is that these individuals have received their good X chromosome from the mother here, but the girls have received one mutated form of the X chromosome from the father, but they're not expressing the disorder because it's a recessive. They have a good copy of the X chromosome as well. But then, if there is mating again between cousins, then this factor suddenly combines with this one to produce both male and females that are affected. Now, the most famous example of this is hemophilia, A and B. And this is famous, among other reasons, because it's a mutation that occurs in the royal families of Europe, including England and other countries, Russia. And this is caused by a defect in one of two proteins, either the clotting factor 8 or clotting factor 9, that's required to make to allow blood to clot. Now, untreated, this is the kind of phenotype or expression that you see. So this is a child who's presumably had just a bump or something, but unlike the rest of us, instead of the bleeding rapidly stopping, the bleeding doesn't stop for a long period of time, and so even small bumps can give rise to very severe injuries. This is a hematoma that resulted just from a bump on the head, and the source that I got this from said that it took 2 months for this to go down. Okay, so 100 years ago, this disease was usually fatal in childhood because most children, have bumps or small accidents, and it was almost invariably fatal. Now it's possible to supply patients with these factors, these protein factors, and so now the life expectancy is almost normal. It's about 10 years shorter than normal, but it's a big improvement. Okay, and then the last pattern that I want to talk about is Y-linked inheritance. And here, the Y chromosome doesn't carry many genes, but it encodes about roughly 50 genes that are active. And most of these have to do with maleness or male fertility. And this would be an example of a mutation here. The star indicates a mutation. In this case, it's one that does not totally block fertility. Otherwise, there wouldn't be any children to look at. But this is one that modifies fertility, for example. And it's passed from generation to generation only through males. And so an example of that is the azoospermia, which is caused by deletions in the AZF regions. There are 3 of these on the Y chromosome. And again, deletions here cause this very severe phenotype where no sperm are produced. So these deletions here, of course, would result in with sterility. So there'd be no progeny. But there are other mutations that are less severe that just reduce the fertility. And then that trait can be passed on to further generations. Okay, so then a couple other things I want to mention about that are important to know about. And that is that not all Mendelian disorders are caused by single genes. And a good example of that is the Charcot-Marie tooth neuropathy that we talked about last week. Again, this is the neuropathy, if you remember the picture, that results in that stork leg, where the lower part of the leg just becomes very skinny because due to loss of nerve and muscle, and the arched foot and the clawed toes. It turns out this is very painful for people. The arching is due to contraction of the muscles, and this is extremely painful for people who have this disorder. Well, it turns out that this phenotype, there's sort of a broad phenotype where everyone sort of meets a certain criteria. When this is looked at more carefully, this phenotype can actually be broken down into more subtle, they can distinguish different forms of this disorder. And it turns out that there are actually 39 loci within the genome that, when they're mutated, give rise to these different forms. So here, instead of having one gene producing one disease, there are actually 39 genes that can produce different forms of the disease. Now, each one of these individually behaves like a Mendelian disorder. So the inheritance of each one, it can be considered sort of independently of the others. They're all rare, so it's unlikely that you get two of them together, although that does happen sometimes. And so this is actually maybe the rule rather than the exception. In other words, that genes that are really disorders that are really caused by only one gene may be rare. And the more common case may be that several mutations in several different genes. For example, if there's a biochemical pathway that's important for something, you can imagine that if mutations in different genes, that encode different proteins in that biological pathway might give rise to the same phenotype. So it's probably the rule that there are more than one gene that can contribute to a Mendelian disease. And that phenomenon is called locus heterogeneity. Okay, the fact is that there's not a single locus that gives rise to a disease necessarily, but there may be many. In this case, there are at least 39. Now, this becomes important later. I think next week I'm going to try to talk a little bit about how genes are mapped, how you find out where a gene that causes a disease is in the genome, and knowing whether a disease is caused by a single gene or multiple genes becomes very important when you're trying to map the genes involved. So this locus heterogeneity is something you need to think about. I don't have a slide for this, but another type of heterogeneity is allelic heterogeneity. And what this refers to is that within any given locus, there may be not one type of mutation that can inactivate that gene, but there might be many. There might be hundreds of different sites within the gene that could be mutated that gives rise to the same phenotype. So in genetics, when you're trying to figure out what the genes are that cause a disease, these are two things that you have to think about, take into consideration. The fact that there may be locus heterogeneity, that there may be scattered loci throughout the genome that give rise to the same disease. And then in addition, for every locus, there might be allelic heterogeneity. So different mutations within the same gene might cause the same defect. Again, these become very important when you're trying to map genes to figure out where the causative genes are located in the genome. Okay. I mentioned before for cystic fibrosis that there are both environmental and genetic factors that can modify the expressivity of the symptoms. And without going into any details at all, these are just a list of genes that have been found to modify different aspects of cystic fibrosis. So these genes, for example, affect the pulmonary function. The ones that are marked in yellow have the largest probability of having an effect. Others may affect the colonization of the lungs by bacteria, but different genes may be important for intestinal phenotypes. So this is the next week's lecture is actually, we're going to concentrate on complex disorders, where actually many different genes are thought to contribute to a single disorder. And you can see, though, that even for Mendelian disorders, which are primarily where there's one gene of primary effect that may control whether you develop the disorder or not. Even there are probably other genes that collectively determine the severity of the symptoms. Okay, so this is, one question is why these genes that cause these horrible diseases, why are they prevalent in the population? Why aren't they just eliminated? And there are two reasons why this doesn't occur. One is that due to something called the so-called the founder effect and in small populations. And that is that if you have a population that's isolated from the rest of the population, and if people tend to find their mates only primarily within the small population, well, if a mutation is brought into that population at some point, it tends to increase in frequency. So a good example of that is the Cajun population in southern Louisiana. Now, you may know a little bit about this. These Cajuns are French-speaking individuals who originally came from French-speaking Canada about 200 or 230 years ago. And there was a migration from French-speaking Canada down to Louisiana, and everyone settled in the same area. And for generation after generation, people in this community tended to, find their mates in the community. And because of this, certain diseases are found at much higher frequency than in the outside population. One of these diseases is Tay-Sachs disease. I'm going to explain what this is in more detail later. But just for right now, just remember the name. It's a disease, actually. It involves it's called the glycolipid storage disorder, where gangliosides accumulate in the brain and cause damage to the nervous system. And the frequency in this Cajun population is again much higher. Well, in this sort of closed community, people have kept very good records, who their grandparents were and their great-grandparents. And investigators were actually able to reconstruct pedigrees that go back over 200 years. This is 13 generations that you're looking at right here of matings. So this is great-great-great-great-great-grandparents. And furthermore, what they were able to do is by sequencing the gene that is known to give rise to Tay-Sachs disorder, they found that the people tended to have the same mutation. And they were able to, everywhere I've put stars, these are people they were actually able to sequence the DNA and show that the mutation was the same. And then that combined with this pedigree that came from, the people's records in the family Bible or whatever, they were able to guess that this mutation here originated way back here, about 230 years earlier, and was actually brought to this region most likely from a person who had immigrated to Canada from France over 200 years earlier. And what you're looking at is this, even though none of these people are very closely related to each other, they have this distant relative in common. Okay, and the mutated gene has come together, and then it causes these individuals to be affected. Now, when this occurs, when one individual gives rise to mutations that then come together later, these mutations are called identical by descent. So this is actually, on the molecular level, the same mutation that this person had up here. Of course, the DNA has been replicated many times in the meantime, but it's actually the same mutation. So the mutations that this person here harbors are identical by descent. They originated from the same person. And as a matter of fact, that's true for this person and this person as well. Now, surprisingly, they found one family where this was not the case, where they found one allele, on one chromosome there was this red mutation, which is the same as the others. But then there was a different mutation on this other chromosome. So this is an example where sort of against all odds, someone from outside of the community maybe, or maybe someone within the community had a new mutation arise. And then those two mutations came together. And so this person has the same disorder, but carries two different mutations. So in this case, we say these mutations or the genes are identical by state. In other words, they're both mutant, but they're not identical by descent. Okay, and so these are two distinctions that become very important, especially when you're trying to map genes to figure out where genes are located on a chromosome. Okay, so again, if a mutation comes into an isolated community, then the frequency of that mutation can become very, very common in the community. And this has been seen for many, many recessive disorders. Now, another mechanism by which mutant alleles can become common is if there's selection for the mutation. Now, there's not usually selection for individuals who have two copies of the mutation, but there are many examples where there seems to be selection for individuals who are heterozygous for that mutation. And one of the best examples, or one that's been studied quite a bit, is Sickle-Cell Anemia, again, Sickle-Cell Disease. And this has been postulated to provide resistance to malaria in people who are heterozygous for the mutation. And this is sort of circumstantial evidence, and this shows the frequency of the mutant allele in Africa, and it compares it to the frequency of infection with malaria. And you can sort of see, for this part of the map at least, there is a good overlap between this mutation and where malaria is. So this provides circumstantial evidence that there might be selection for this mutation in this area. Now, up in this region here, there's also malaria. It turns out there's a different type of disorder that affects hemoglobin called alpha or beta thalassemia. And this has also been proposed. People who are heterozygous for these thalassemias have also been proposed to be resistant to malaria. So it could be that resistance to malaria in this part of Africa, this actually holds for the Middle East in general, that in the Middle East, resistance to malaria may have promoted increased the frequency of these thalassemia mutations, whereas in sub-Saharan Africa, it may have increased the frequency of the sickle-cell anemia mutation. And here's just a list of some diseases where this has been looked at. So sickle-cell anemia and thalassemia is just what I talked about. This is a very interesting gene. This glucose 6-phosphate dehydrogenase deficiency results in the hemolysis or hemolysis of destruction of red blood cells when people take certain drugs or when they eat certain things. And so this was first discovered during World War II when its troops were given a drug called primaquine. This is an anti-malarial drug, and the troops were given, hundreds of thousands of troops were given this drug to help prevent malaria infection. And it turned out that many African American troops developed very severe anemia. And when looked at more carefully, they found that the drug was causing the red blood cells of the troops to lyse, and so they became anemic. It was also found in certain populations that eat a bean called the fava bean, that this can also induce hemolysis as well. And it turns out that the reason for this is that this glucose 6-phosphate dehydrogenase is part of the pathway that makes NADPH in the red blood cells. And NADPH is required to reduce something called glutathione. And reduced glutathione is a molecule that sops up free radical ions. So it's part of a mechanism for preventing oxidative damage due to free radicals. And so people who are deficient in this are deficient in the glutathione. So they can't sop up the free radicals as they should. And those free radicals then damage the cell membrane of the red blood cells. and that causes the cells to lyse very easily. So it turns out it's not only primaquine and fava beans that do this. There's a list of like 30 different chemicals, drugs that are a problem for people who have this deficiency. So this is an example of sort of a new area of research that's emerged in the last 30 years or so. It's called pharmacogenetics or pharmacogenomics. And it is a field of research that involves looking at the effect of the genetic influences on the effect of drugs. So in the future, you're going to be physicians and you're going to be prescribing drugs. And more in the future, you may have to take genetic information into consideration when you make those prescriptions. So for example, right now, if you were a doctor, you would have to be very careful about prescribing this drug to African American patients, because there's a certain probability that, there's almost a certainty that a certain fraction of those individuals would suffer severe anemia. They might even die from it. So the understanding how the genetics of a person changes the response of that person to the drugs that they're given is now already an important part of medicine, and it's going to become even more important as more and more of these relationships are understood. Okay, so then cystic fibrosis we've talked about. This is a little less certain than these studies, but there's a theory that this individuals who are heterozygous for these mutations may be more resistant to cholera and tuberculosis. This disease, by the way, is found at very high frequency among the so-called Ashkenazi Jewish population in Northern Europe. These are a group of Jewish individuals who live in Germany and German-speaking parts of Poland and Czechoslovakia. And together, this is called the Ashkenazim. And cystic fibrosis, there's a deletion of phenylalanine at 508 position that is one of the mutations that causes cystic fibrosis. And this is found at very high frequency in this population. So, and also Tay-Sachs is very frequently found in that population as well. It's the same mutation that we talked about in the Cajun population. And very interestingly, people, because of the high frequency of these dangerous alleles in the population, and also the tendency for people within the population to marry within that community, there's a very proactive measures are taken. In other words, when two people want to get married in this community, they usually go to a genetic counselor. They get tested, and then they find out whether they carry these mutations or not. And then the genetic counselor talks with them about whether it's really a good idea to get married, or if you get married, you have to take these precautions. And because of this sort of proactive stance, the frequency with which you find patients with cystic fibrosis or Tay-Sachs disease is decreasing in the population. People are becoming aware of and not just letting this develop by chance. People are being proactive about taking measures not to have children that have these disorders. Okay, so then let's see. Okay, so the next thing I want to talk about are different types of mutations that occur. And we'll talk about point mutation, indels, and repeat expansions. So point mutations, again, are ones that just change one base pair. And even though it's just one base pair out of hundreds or thousands within a gene, this can have big consequences. So we've already talked about sickle-cell anemia. This is a change in one amino acid from, I think it goes from glutamate to valine. Yeah, I think it may be hard to see, but here, this is glutamate here, and this is valine. Oops. And that is enough to change the charge. So I think I said it wrong before. This would have a negative charge, one more negative charge compared to valine, which is not charged. And so this changes the electrophoretic mobility, and that's what Pauling detected in his experiments. And it also changes the conformation of the hemoglobin molecule, and this causes it to stack in inappropriate ways and makes clusters of molecules inside the cell that then actually changes the shape of the cell. So even the change of one nucleotide can have a huge effect. In addition to that, there are nonsense mutations that introduce a stop codon in the middle of a gene. Now, interestingly, the effect of this can be quite different in different situations. So if you have a protein like this and you stop the expression or stop the translation in the middle, well, sometimes that has almost no effect on the protein. Maybe if this part is the active part of the protein, if you lose a little bit, it doesn't make any difference, and it's still active. In other cases, though, in some cases, though, even if the activity of the protein is not, the intrinsic activity is not destroyed by that stop mutation, it can still eliminate the expression of the protein. And this occurs by a pathway that was just discovered sort of relatively recently, and this is called nonsense-mediated RNA decay. And what this means is this phenomenon involves the idea that if you make a peptide that's been prematurely terminated, the total mRNA may be degraded. And if that occurs, then you get no protein expressed. And the way this works is this way. You have the mRNA comes out of the nucleus in the spliced form, right? In the nucleus, you have the exons are brought together and the introns are eliminated. And then this spliced messenger RNA comes out of the nucleus. But when it does so, some of the splicing factors are still bound to it. And what happens then is that when the ribosome starts to translate that message, it systematically displaces those proteins during the first round of translation, right? So this is the 5' end of the message, this is the 3' end of the message. This ribosome is going to recognize the first appropriate AUG codon, put a methionine in, and then start moving down the transcript. And as it does so, it displaces these splicing factor proteins that are still stuck to the message. Well, what happens is if it hits a stop codon, that causes the ribosome to dissociate from the mRNA. And what you're left with then is an mRNA molecule with these residual proteins still left on it. This complex then becomes a signal to degrade this message. And so the entire message is destroyed. Okay? So this displacement occurs on the first translation of the message. And so if it's destroyed, there's no message to translate anymore, and essentially get no protein expression. Even though the protein, if it were made, would be still active. Do you see that? So the effect of the stop codon may not be, it could be to make an inactive form of the protein, but in addition to that, it may destroy the messenger RNA and result in no protein being expressed. An example of this is here, where stop mutations that are found in this PAX10 gene here that are early exons make a peptide that's much smaller, and this results in the message being destroyed and a very severe phenotype because this is just not being made. Whereas stop codons that occur later in later exons, stop codons in the last exon are immune to this. So stop codons in here actually don't trigger this nonsense-mediated decay, and so peptides are made. In this case, though, it results in peptides that have gain of function and probably produce proteins that cause other problems. So where the stop codon occurs can have two very different consequences. Okay, so another ways that mutations can affect gene expression is by changing RNA splicing. And so here, for example, the acceptor sequence for the intron is, I think it's GT is GT is the normal acceptor sequence. But if that's changed, then splicing does not occur at this point, and you get inappropriate expression and probably, a nonsense protein being made. In this case, a mutation can sometimes create a splice site that's an inappropriate splice site, and then make the splicing inappropriate, and then give rise to a non-functional protein. Let's see, here, there are splicing factors that bind to the exon as well as to the joint between the exon and the intron. Within the exon, there are binding sites for proteins called exonic splicing enhancers. And so if mutations interfere with the binding of those enhancers, that can also change the splicing of the mRNA and result in non-functional gene. Okay. So it's not just changing the codon sequence that's important. There are many different ways that point mutations can interfere with gene expression, and these are just sort of several examples. There are other ways as well. Another way that point mutations can affect gene expression is by changing the behavior of the promoter region. So the promoter is a region where RNA polymerase II binds and many other factors bind in that in a place where transcription is initiated. And if point mutations interfere with the binding of a needed transcription factor, that can result in inappropriate or no expression. A very interesting example is this Clotting Factor 9 that we talked about earlier. So when this is defective, it causes hemophilia in a recessive manner. And it turns out that there are two different classes of mutations that have been found in the promoter region for this gene. And the ones with the dark arrows here are ones that interfere with gene expression, but only in children. It turns out that the way this gene is regulated changes during development. And so these sites right here interfere with the binding of factors that are required for gene expression in juveniles, but in adults, they're not required. And so people with this are carry one of these mutations, have hemophilia when they're young, but then they grow out of it. When they grow up, they don't need, they're not hemophilic anymore. In contrast, people who have this Brandenburg mutation right here lose expression for their entire life. So here's another example of how different point mutations can modulate gene expression and result in different phenotypes. Okay, now this is another type of mutation is involving the insertion or deletion of several nucleotides, and the effect of this is often to change the reading frame of the gene. So if this occurs in a codon reading frame, so for example, in Tay-Sachs syndrome, there's an insertion of 4 nucleotides in the reading frame for this gene, and it causes the coding for inappropriate amino acids and eventually a stop codon. And then this is the cause of the Tay-Sachs syndrome. This is the common mutation that's found in that Cajun population and also in the Ashkenazi Jewish population. These are, we've talked about point mutations and also indels, small insertions, deletions. Last week we talked about the fact that the age of the mother was very important for determining the risk of trisomies, especially trisomy 21. And you saw a very dramatic increase of trisomy 21 and other trisomies with maternal age. And I promised you to talk to you about problems with the fathers. And this paper was just published a month or so ago, which looks at the de novo mutations that are passed from the father to the child as a function of age of the father. So at a very young age, the father is transmitting roughly about 30 to 40 new mutations to the progeny, to each of his progeny. So each of you got that many, at least that many from your father. And as you can see, as the age of the father increases, there's a dramatic increase, at least a doubling of the number of mutations. So fathers who are more advanced in age may be giving 90 or 100 mutations to their progeny. And even more advanced, it may be even twice that much. And so this has been discovered recently. It's actually been known for a while. And it's not too surprising given the fact that men are producing sperm continuously through their life. So there are many replications involved. And every time you have a replication, there's always a small chance of creating a mutation. And so there is definitely an increase in the number of mutations that fathers pass on to their children as they grow older. And it's also interesting that the incidence of schizophrenia and autism also increase with the age of the father. So there seems to be some connection there. Okay, now the last type of mutation that I want to talk about are, this is a very interesting class of mutations called repeat expansions. And these are famously found in, two of the most well characterized as Huntington's disease and Fragile X syndrome. And what this involves is an expansion of triplets, repeats. So in Huntington's disease, there's a CAG that is found within the coding sequence of the Huntington gene. And this normally, in normal individuals, this is found, it's a repeated sequence, it goes CAG, 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 and the number of repeats is usually significantly less than 40. If it's greater than 40, the protein that is produced becomes pathogenic and gives rise to a very terrible neurological disorder called Huntington's disease. In Fragile X syndrome, there's a repeat of CGG. This is found within the 5' end part of the gene of the mRNA. And here what happens is when this expands, you get excess methylation. Remember, this is a CPG right here. And by expanding that, the cell decides to methylate it, and so it turns off gene expression. It inhibits the gene. So the protein, the coding sequence has not been changed at all as a result of this mutation, but the expression is dramatically downregulated. So in the case of Huntington's disease, here's what happens. This is a dominantly inherited disease. This is just one of the most terrible diseases you can get. This is called the striatum of the brain. This is a coronal section, so you're looking, it's cut like this, and you're looking in this direction. And this is the striatum right here. And you can see that this part of the striatum is just totally absent from the brain of a person with Huntington's disease. This is a disorder that people don't become symptomatic until middle age, usually, until they're in their 40s or 50s in many cases. And it's invariably fatal. And so if there are genetic tests now to see whether you carry a lethal mutation or not. And if you choose to have this test and choose to find out the result, you will know with certainty whether you're going to die a horrible death. It's really, no one escapes from this disease. This is invariably fatal. This is, the way the diagnosis is made is that it's possible to, have you studied PCR yet? Do you know what that is? You know what PCR, polymerase chain reaction? You're going to do this in one of your labs. So just right now, it's a technique that allows you to take a specific part of the genome and amplify it and then look at it. Okay. And so what they've done is they've subjected the Huntington gene to this PCR protocol, and then they're looking at these amplified sequences. And they can tell how many repeats there are by analyzing the mobility of these fragments by gel electrophoresis. So the normal allele length is right here. Okay, and this is right at the edge where where the length becomes pathogenic. You see this is from this individual here who had already died. And these are the lengths that you see for the children. So these are 3 affected individuals and someone who will become affected. He's just not, it hasn't appeared yet. And you can see that the lengths of these expanded, repeats are larger. And so it turns out that there's an inverse correlation between the length of these repeats and the age of onset. So the longer the repeat you have, the earlier the disease strikes. And that's shown in the graph here with this showing the variability. Let's see. The mechanism, let me point out one thing. I'm going to talk about this again in just a minute, but you notice that the children all have longer lengths than the original parent right here. And this is a phenomenon that is observed for all of these triplet repeat disorders. And it's called anticipation, where the first person to be affected usually has the mildest form of the disease, and their children have a more severe form, and the grandchildren have even a more severe form. And I'll show you why this is in a minute. And that phenomenon where with every generation the disease strikes earlier and is more severe is called anticipation. Now the mechanism underlying this is thought to underlie this, during the replication of this part of the DNA that contains this repeat, it's thought that the newly synthesized strand may disassociate and then reassociate inappropriately, and forming this kind of template for DNA polymerase to work on. And you notice this is sticking up in the air, so when this is added, instead of having 3 repeats now, you have 4 repeats. And it's thought that in these regions where there are many, many repeats, that during DNA replication, there's this slippage. There's synthesis and then reannealing, and then synthesis and reannealing. And that process makes it longer and longer. Here's a study that was done. This is actually done by someone, the professor's an acquaintance of mine from Johns Hopkins, where they looked at, everyone the black circles and squares are people who have developed Huntington's disease. And what you're looking at are the age of onset and then the number of repeats. So in the first generation, the age of onset was 34. Among those children, it's not roughly the same. It's 35, 40, I can't read that, 39, 35, 33, what is that? 38. And then these are the number of repeats. So there's 51 repeats, 48, 50, 50, 40. And then, but if you look at the next generation, the onset is earlier. This one, the onset's 30 with 47 repeats. Onset at 18 with 82 repeats. And then onset of 22 with 64 repeats. And onset of 21 with, I can't quite read it, 60-something repeats. And then you go to the next generation, the onset is 9 years old with 78 repeats. So this shows very clearly this phenomenon of anticipation. With every generation, there's a tendency for the repeats to be longer and the onset of the disease to be earlier. And it's thought that it's from the slippage mechanism. Okay, so then it turns out there are many, many Mendelian disorders that have been analyzed now. Just within the last 10 or 15 years, approximately 3,000 Mendelian disorder genes have been identified. And the remaining 1,000 or so that are known will probably be dissected within the next few years. The next big challenge, however, is trying to understand complex disorders, which are much more common. So most of these diseases are very rare in the population. Complex disorders, by contrast, obesity, diabetes, heart disease, mental illnesses like schizophrenia, these are severe depression. These are much more common disorders and are probably the result of not individual genes, but the collective activity of many genes. And so the current challenge for molecular geneticists is to figure out what those genes are and how they interact. And then based on that, what kind of preventive measures or corrective measures can be taken. Okay, so I think we'll stop here. There are 3 more slides that have to do with prenatal diagnosis. You can just look at those. Someone asked me last week about, what's possible. And you look those slides over. It's also discussed in the textbook, and you can get some information there. Okay, so next week it's going to be complex disorders.